Okay, we're live na po.
Now you can
Admit
let
all
everyone in. Admit all. It's 7:55.
We're live na po.
Science and technology and innovation, these are indispensable in the modern economy, in the modern world.
Meeting at the MSI, and we resolved among about 6 to 8 of us, local members, first to form PAASE Philippines so that we could continue to host meetings here, but also to go on a massive campaign to try to get support from the national government through the legislative and executive branches to invest more in science and technology. So it started with a white paper, a position paper that we co-authored among 6 of us, namely Ernie Pernia, Cesar Saloma, Redor Asansa, Toby Dairita Fataneo, and Alvin Colaba de la Salle, and myself.
So PAASE historically, in the past 10, 20 years for sure, have been collaborating with our counterparts here in the Philippines at various universities. And a lot of these interactions probably started as peer-to-peer. But thankfully, we have the Department of Science and Technology here in the Philippines, and they've come up with some really helpful and very impactful programs that actually enable PAASE members that are based in the United States and other parts of the world to actually come here and do extensive research and academic partnerships and collaborations.
Well, I think that one of the good things that PAASE is doing is really trying to stay in touch with the researchers and the scientists here in the Philippines. So I think that is one of the good programs that they keep on supporting some of the initiatives of our researchers and scientists here in the country, trying to align also their assistance towards the plans of the Philippine government.
It's very important that the PAASE members and scientists based in the Philippines are continuously in contact, so we will know the needs of the Filipinos and opportunities that Filipinos can have to do science with their PAASE counterparts. So these interactions, such as the conference that we're holding and the annual conferences that we have.
The Philippines and the scientists there have very unique perspective that adds to discoveries and inventions advancing in general the field.
These members, because they are part of an academy, it implies that they are productive. So they have publications, they have innovations, they have inventions, patents, and they have, they have an impact on society. Okay, so these are the people who can convince government, invest more in science and technology, and that was really the gist of our paper, our position paper in 2000. And it is Ernie Pernia who said that R&D by nature is expensive and it is a public good. So it is important for the government to take the lead in investing in R&D.
To increase the economic growth potential of the country, we cannot continue to just rely on traditional inputs into production as capital and labor. We also need to develop our expertise in science and technology and innovation so that we can come up with new systems, new ways of doing things.
I think that science, researchers, industry, and even the government is really like an ecosystem of knowledge. So I think it doesn't really matter if you're coming from a private institution or a public institution, such as a state university. I think we all have a role to play to improve the research capabilities and scientific knowledge here in the country. I think it is about time for us to realize that one of our roles as academics, as researchers, as scientists, is to actually contribute to that global body of knowledge.
And the role of this cooperation among these government agencies really is to be able to attract science and technology, global-based companies to set up shop here. And that is really key in terms of developing the science and technology innovation ecosystem in the country.
Particularly because there's that deep desire. And what PAASE does and can do more of is to promote this relationship between its members.
We really need to ratchet up the appreciation, the recognition of the importance of science and technology and innovation.
And we are all trying to attain what we call the common good, the greater, higher, long-term good of the Filipino people.
Good morning, good evening, good afternoon, wherever you may be. Welcome to the Philippine American Academy of Science and Engineering 40th Anniversary and 2020 Annual Scientific Meeting that is running for 4 weeks. We began yesterday with an excellent plenary session, and today we have the first parallel sessions. This one is parallel session 1.3, Disease, Medicine, and Pharmaceutical Sciences, that was indefatigably organized, as was the rest of this entire meeting, by the real power behind PAASE, our president, and my co-host for this morning, Dr. Gisela Concepcion. I think you're muted, yeah. Giselle?
Good morning. Good evening, everyone.
This is Giselle. Thank you, Joey, for that nice introduction. Welcome, everyone, to Parallel Session 1.3 on health. And we're happy to have a very special lineup of speakers today. And we're hoping that this will be a very stimulating session. So before we start with our first speaker, we'd like to just do two little plugs because this is our 40th PAASE anniversary. So we're hoping that our members will show full support to our organization. And we came up with this Fresh Start campaign for PAASE Lifetime Membership Dues. And we're trying to raise funds because PAASE has many worthwhile projects that we need to support. And we thought that it'd be best for our members, well, not to worry about annual dues. So why not do the lifetime membership due? And if you had forgotten to pay it in the past, then there's some kind of an amnesty with this Fresh Start campaign. Okay, so it's easy to pay this now. There's PayPal, and then we have our local bank account. And then it's timely because it's a 40th year and we have an online conference, which does not have a registration fee. So the amount of the lifetime membership dues for Filipino members is about the same amount. So help us raise some funds for PAASE so that we can pursue our project. So I'll move to the next slide. So Richmond Pancho, our co-host and the webmaster, will show us the slide for donations. So we also have a donations campaign for all members who find other donors, including companies that they are connected with. And with that donation campaign, we would really get a lot more funds. And this donation campaign is going to take off because we have tax-exempt status now in PAASE. So 100% tax deduction of your donations. So I think that's enough for plug. And now let's go for the real thing. Perhaps before this, let's take a screenshot, everyone. And that's because some of our speakers might have to leave. They have other important meetings after their talks. So let's do the screenshot now. Richmond, are we ready? Ready, set, go.
Maybe request everyone to please turn on their webcams, please. Thank you.
Please turn on your webcams if you can. Okay. Okay. Thank you.
We're still not ready to. Okay. All right.
Okay, ready?
On my mark. 1, 2, 3, smile. Okay. Next batch, we have 2 windows.
Okay.
Next window. Ready. Hold on. I need to save the first photo. Okay. All right. Next window. Ready. 1, 2, 3. Okay. Thank you.
All right. So now we're ready, and I am to introduce our first speaker. So our first speaker is Homer D. Pantua, DVM PhD. Homer is a principal scientific researcher at the Department of Infectious Diseases at Genentech, a biotechnology company which pioneered the development of therapeutics from recombinant DNA technology. He has led both antiviral and antibacterial discovery efforts and is now serving as a virologist for Genentech's COVID-19 exploratory research. Dr. Pantua obtained his PhD in biomedical sciences from the University of Massachusetts Medical School and is Doctor of Veterinary Medicine from the University of the Philippines, Los Baños. He initially received infectious disease training at the Research Institute for Tropical Medicine in the Philippines. He has maintained his connection to the Philippines by being a Balik scientist in 2011 and by founding BioAssets Corporation, a veterinary research and diagnostic company, in 2014. His passion is to develop therapies and vaccines for infectious diseases in humans and animals as well. Homer?
Yes. Yeah, thank you for a very generous introduction. I would like to thank the organizers of the annual PAASE scientific meeting and symposium for giving me the opportunity to share the science that we do. I am Homer Pantua, an infectious disease scientist at Genentech in South San Francisco in California. Here you can see the entire Genentech campus along the San Francisco Bay. So what I'm going to share with you today is one of our antibacterial drug discovery project, which focuses on the unstable mechanism of resistance to Escherichia coli lipoprotein signal peptidase. Just to give you a high-level summary, I hope to show you through medicinal chemistry efforts that we have identified a potent globomycin analog G0790, and that inhibition of LSPA leads to the accumulation of peptidoglycan-linked LPP, which we believe as the toxic intermediate. In addition to the previously described resistance mechanism to globomycin, which is LPP deletion, we also uncovered two other mechanisms of resistance, such as downregulation of LPP, heteroresistance phenotype mediated by genomic amplifications of LSPA that result in increased LSPA levels sufficient to overcome inhibition by G0790 in culture. And that these genomic amplifications are highly unstable and are lost after as few as 2 subcultures in the absence of G0790. The World Health Organization categorized Enterobacteriaceae, which includes E. coli, Klebsiella, and Enterobacter, and the non-fermenters Pseudomonas and Acinetobacter as the first priority antibiotic-resistant pathogens. These bacteria are major pathogens in urinary tract infection, hospital-acquired pneumonia, and intra-abdominal infection. Globally, about 700,000 deaths each year are estimated to be attributed to resistant infections. And if left unaddressed, global burden of deaths could reach 10 million every year by 2050. This schematic shows the chronology of antibiotic discovery, starting with penicillin in the late 1920s. This also shows the golden age of discovery and the golden age of medicinal chemistry. I'd like to point out, however, that for the past 40 years, there is no new antibiotic with novel mechanism that has been discovered. What is concerning is the rapid emergence of resistance to these antibiotics. What is even more concerning is that as the antibiotic resistance rapidly increases, more companies are stopping antibacterial discovery efforts due to development hurdles. Beside the development risks, antibiotic development is not an attractive investment proposition for private capital. Antibiotic lacks the financial upside of other products such as in oncology, respiratory diseases, dermatology, musculoskeletal diseases, cardiology, and hematology. However, in keeping with our company commitment, which is doing now what patients need next, Roche and Genentech is committed to take on the innovation challenge of the AMR Action Fund and will continue to support antibacterial discovery efforts to address AMR. Here is a cartoon of an E. coli showing the flagella, the pili, and the chromosome in the cytoplasm encased in bacterial membrane. The gram-negative membrane is an asymmetric membrane that has an inner membrane, an outer membrane which contains lipopolysaccharide, separated by a periplasm which containing a peptidoglycan layer. Antibiotics like vancomycin and rifamycin are not as effective in wild-type E. coli. The minimum inhibitory concentration, or the amount of drug needed to completely stop bacterial growth, is decreased in sensitized strains or strains that have disrupted outer membrane, which suggests that the gram-negative outer membrane is a permeability barrier. Bacterial lipoproteins play an important role and diverse roles in bacterial physiology and pathogenesis. It is proposed that at least 90 lipoproteins in E. coli are mostly localized in the outer membranes, and certain lipoproteins and the enzymes involved in their biosynthesis and localization are essential. The lipoprotein biosynthesis pathway involves three enzymes, LGT, LSPA, and LNT, with LSPA and LNT being crystallized in-house. LGT is a transferase that catalyzes the transfer of diacylglycerol from phosphatidyl glycerol to the cysteine of the lipobox of the pro-lipoprotein. On the other hand, LSPA is a signal peptidase that cleaves the signal peptide. Lastly, LNT is another transferase that catalyzes the transfer of the third acyl group to form the mature lipoprotein. To determine whether LSPA is an attractive drug target, we assess the essentiality of LSPA in bacterial growth. This graph is an in vitro growth assay where y-axis is CFU per mil versus time in the x-axis. The black line represents the wild-type E. coli, and the red line represents the LSPA knockout. Deletion of LSPA resulted in bacterial cell death, suggesting that LSPA is essential for growth of bacteria in vitro. Complementation of the LSPA knockout with an Acinetobacter LSPA rescued the growth in vitro, suggesting that the conservation of LSPA across multiple gram-negative strains. We also showed that the lipoprotein biosynthesis pathway is essential for growth in vivo. As shown here in a bacteremia model, we show that the LGT knockout and the LNT knockout did not grow compared to their wild-type counterpart after 24 hours of infection in a neutropenic mice. What we hope to develop is a potent inhibitor of LSPA. And our therapeutic hypothesis is that inhibition of lipoprotein synthesis will mislocalize and or deplete essential and virulence-associated lipoprotein, leading to bacterial cell death. Another reason that LSPA is an attractive drug target is the availability of a known chemical matter that inhibits its function, and that is globomycin. Globomycin is a natural product isolated from Streptomyces species. Globomycin is comprised of 5 amino acids cyclized with a beta-hydroxy acid lipid tail. Globomycin also inhibits LSPA with MIC against wild-type E. coli. However, globomycin is not a suitable clinical antibacterial candidate due to inefficient penetration to the impermeable outer membrane. With the goal of increasing globomycin whole cell potency through medicinal chemistry optimization, we identified G0790 as an analog with increased growth inhibitory activity as measured by MIC. And as you can see here, about the 16-fold improvement from globomycin to G0790. G0790 contains a diaminopropionic acid, DAP, a cyclohexylglycine, and an N-methyl norvalin residues in place of the serine, isoleucine, and leucine residues, respectively. Molecular modeling based on the crystal structure of LSPA in complex with globomycin suggests that the DAP engages the two catalytic aspartate residues. We confirmed that the increase in potency against wild-type E. coli was specific to LSPA using multiple parallel approaches using a previously characterized resistance strains deletion LPP and a recombinant highly expressing strain LSPA. As you can see here from 3.1, the MIC shifted to 25, suggesting a specific activity against LSPA. Serum MIC has also improved, suggesting that there is a decrease in serum protein binding. MIC with sensitized strain and biochemical IC50 is comparable between globomycin and the G0790, suggesting that the improvement on G0790 is due to penetrance. To determine if the increased wild-type E. coli activity was driven by decreased efflux, we tested G0790 in a deletion strain which lacks the outer membrane protein TOL-C component of the major efflux pump. So the wild-type MIC versus the TOL-C MIC shift for globomycin and G0790 is approximately 31 and 4-fold shift respectively, suggesting that the reduced efflux contributes to the improved wild-type E. coli activity. Inhibitory activity also improved in multiple gram-negative strains, but not to the gram-positive Staphylococcus aureus USA 300, suggesting a less likelihood of off-target activity. We also show that G0790 has bactericidal activity as shown in the time kill assay, where you can see that the wild type grew while bacteria treated with G0790 significantly decreased the CFU to the limit of detection. This time-lapse microscopy also showed the progression of swelling of bacteria that leads to the bursting and cell death. We then look closer to what is happening in the membrane by electron microscopy, and we found that there is accumulation of membrane upon treatment with G0790. And from a membrane fractionation data that I won't be able to show you today, inhibition of LSPA leads to the accumulation of peptidoglycan-linked LPP, which we believe to be a toxic intermediate. And it also leads to the depletion of essential lipoproteins. To determine the resistance profile to G0790, we performed a resistance selection study, and this involves 10 independent cultures grown in the presence of G0790 at different concentrations over MIC. We then recover the CFUs and calculate the frequency of resistance of those isolates. What we found is that G0790 has frequency of resistance that is within the acceptable range, which is around 10 to the minus 8. And the MIC shift of these resistant isolates is about 8 to 32-fold increase. We further characterize the resistant isolates by looking at protein expression of LPP that forms the toxic intermediate and the LSPA. We also, so as you can see here, you can clearly see that in a Western blot analysis, LPP, looking at LPP, LSPA, and a loading control, the LPP is absent in resistant isolate 4 and 9. And by whole genome sequencing, we found that there is a large-scale deletion of LPP, which is around 12 to 22 kb scale, suggesting that the bacteria has developed a mechanism to resist treatment with G0790 by deleting LPP and decreasing the toxic intermediate. Furthermore, we also observe a downregulation of LPP expression, and by whole genome sequencing, we found an insertion of insertional elements ISEC10 and ISTB upstream of LPP and downstream of LPP. And these insertional elements confers resistance to G0790 by downregulation of the LPP, which causes the toxic intermediate. Furthermore, we also look at the protein expression of LSPA. As you can see here, there's a modest increase in expression compared to its parental control. And by whole genome sequencing, what we found is that in these whole genome sequencing results, you can see here in green the location of LSPA, the parental baseline in here below, and this big plateau of amplification contains LSPA and the surrounding genes. So resistance is not good for a pipeline perspective. So we therefore assessed the virulence of these G0790 isolates. Sensitivity to serum is a good indication of attenuation. We therefore subjected the wild type and the resistant isolate in a serum killing assay, which reuses normal human serum, which contains complement factors, and heat inactivated serum, which kills the complement factors. All G0790 resistant isolates are sensitive to normal human serum, except for the genome amplification isolates. Although genome amplification isolates are not highly sensitive to serum, the resistant mutants are highly attenuated in vivo in a bacteremia model using a neutropenic AJ mice. So we further characterize the genome amplification resistant isolates by assessing the stability of the resistance property. So in this experiment, we cultured the genome amplification isolates and passage in broth, either in the presence or absence of G0790. So this graph shows the MIC of parental and the G0790 resistance strain after day 3 of passaging. The first panel shows that in the absence of G0790 pressure, the MIC of this strain reverted to levels comparable to the parental strains. In contrast, while maintaining the MIC of these resistant isolates in the presence of the G0790 pressure. In contrast, the resistant strains which contains insertion elements and deletion of LPP were not affected. And this data is supported by the whole genome sequencing result of these passaging experiments, where we found that genomic amplification were decreased or lost in the absence of G0790 while being maintained in the presence of G0790. So heteroresistance is a phenomenon in which a small subpopulation transiently displays a substantially higher MIC. It has been shown that the amplification of the AMC gene also confers resistance to beta-lactam. However, heteroresistance should be differentiated from persistence. And persistence is the ability of a small population of bacteria to survive bactericidal concentrations of antibiotic. And to determine whether the genomic amplification or LSPA amplification is a heteroresistance phenotype, we perform a population analysis profile, which is a gold standard to determine heteroresistance. And in this assay, we enumerated the CFU from a serial dilution of bacteria grown in increasing concentrations of 0790. And heteroresistance is defined if there is a greater than 8-fold difference between the highest non-inhibitory concentration and the highest inhibitory concentration. As you can see here in these graphs, this shows the ratios of the CFU and the total CFU per plate. And looking at these three strains with genomic amplification, we can see that there is a greater than 8-fold difference between the highest non-inhibitory concentration here, shaded in gray, and the highest inhibitory concentration, suggesting that the genomic amplification with LSPA is a heteroresistance phenotype. To further characterize the heteroresistance phenotype, we check if there is a heterogeneity in copy numbers within a given GA isolate. And indeed, by qPCR, there is an increased LSPA copy numbers and with heterogeneity in nature. As you can see here, compared to the parental strain, there is about 2 to 3-fold increase in LSPA. And in these isolate, there is around 2 to 8-fold increase in LSPA. And this increase in copy numbers translate to the upregulation of the LSPA gene quantified by qPCR in these isolates relative to the parental. This upregulation of the LSPA gene is translated into modest increase in LSPA protein levels in the inner membrane of the in either the wild type or the UPEC deletion LPP. And taken together, these data suggest that the unstable heteroresistance caused by genomic amplification is mediated by modest upregulation of LSPA protein levels in the inner membrane of the E. coli. And as a summary, I've shown you that these genomic amplifications are highly unstable and are lost after as few as two subcultures in the absence of 0790. And that there is a high risk of being misclassified as susceptible by routine antimicrobial susceptibility testing. And this emphasizes the importance of considering the potential impact of unstable and heterogeneous phenotypes when developing antibiotics for clinical use. With this, I would like to thank the Infectious Disease Department, the Medicinal Chemistry Department, Computational Chemistry, CROs from China, Biochemical and Cellular Pharmacology, Structural Biology, Translational, Research Pathology, Safety Assessment, DMPK, and Early Development Biochem. And these are the many people who help us get to this point and bring this close to 3 years of work. With this, maraming salamat po, and I would take any questions.
Thank you very much, Homer. We will reserve questions for after the first batch of speakers have presented. I would like to now introduce our next speaker. Juan Lorenzo Pablo is a molecular biology and biotechnology alumnus of the University of the Philippines, Diliman. He completed graduate studies at Duke University under the mentorship of Jeffrey Pitt and postdoc training with David Clapham. He is currently a senior scientist in Anna Greca's group at the Broad Institute of MIT and Harvard to speak on inhibition of TRPC5 as a therapeutic strategy in kidney disease. Let us welcome Juan Pablo.
I'd like to begin today by first thanking the organizers for this wonderful opportunity to share some work that I'm really excited about, revolving around the ion channel TRPC5 as a novel therapeutic target in kidney disease. We find that there are many compelling reasons to work on kidney disease, chief among this the enormous and growing unmet clinical need in this space, especially as the rates of obesity and diabetes rise throughout the world, conditions that we know lead to kidney complications. And even though the kidney space has lagged behind others in terms of finding molecular targets for therapeutic intervention, we have gained significant insight into kidney biology by studying patient populations that harbor rare genetic mutants and present with kidney disease. Many of these mutations impinge upon a pathway that regulates the cytoskeleton of these cells and is characterized by the nodal signaling molecule RAC1. And through years of research that I won't have the time to go over through today, we have found that the ion channel TRPC5 is an important regulator of this RAC1 pathway. This is underscored by work in rodent models of kidney disease. So we see here in this top panel of images, immunofluorescence images that show us that TRPC5 is expressed in structures of the kidney called glomeruli that are delineated by this glomerular marker synaptopodin. So you see TRPC5 nicely colocalized with synaptopodin here. These glomeruli are comprised of a few different cell types, but chief among these is a population of cells called podocytes. And these cells are so called podocytes because they have these cytoplasmic extensions that are called foot processes. And you see these foot processes here in this electron microscopy images in these lower panels. So in a normal condition, this is what they look like. But when the kidney is exposed to an injurious stimulus, such as lipopolysaccharide here or protamine sulfate, we ablate these structures in a process called foot process effacement. And what's important is that when we genetically delete TRPC5, we are able to prevent this foot process effacement. So we're able to preserve the integrity of these podocytes. And that's shown here in the lower panels where KO refers to TRPC5 knockout mice. So what is TRPC5? Well, the gene TRPC5 is part of a superfamily of TRP channel encoding genes. And the protein product of the gene TRPC5 encodes this plasma membrane molecule that creates a pore in the membrane that will connect the external compartment to the cytoplasmic interior of the cell upon a certain stimulus. In this case, usually G protein coupled signaling. And for those of you who are GPCR nerds, in particular GQ11 signaling. Now, the key metric by which we usually measure ion channels is through the gold standard method of electrophysiology, where we record the electrical activity that is a result of channel opening and closure. And we usually express the relationship of current and voltage in the cell through these graphs, which we call current voltage curves. And what's important for the rest of the talk is that different channels have different current voltage curves. And so in many cases, we can really characterize a channel and know its identity based on the particular shape of the current voltage curve. And for TRPC5, this is a very characteristic doubly rectifying shape. And all that really means is that you have this curve downwards and this curve outwards. And that's all you really have to remember for the rest of the talk. So 3 years ago now, our group found a molecule that is able to inhibit TRPC5. So even though we showed that genetic deletion of TRPC5 is protective against kidney disease, obviously genetic deletion is not currently a viable therapeutic approach, which really led us to pursue small molecule inhibitors. And so we found this molecule called AC1903, which inhibits TRPC5 selectively over very related family members, TRPC4 and TRPC6. And you see here again those glomerular structures that I talked to you about. And so this is what it looks like in a normal wild type condition. If you overexpress the angiotensin receptor in the podocytes, you get kidney damage, as shown here by the cyst-like phenotype in this glomerulus. This is reversed or prevented by co-administering AC1903 to these rats. Now, this is obviously a great proof of concept, but AC1903 is not quite ready to be injected into a human, for example. It doesn't have all of the drug-like properties that we would need. In particular, we would prefer for it to be more potent as well as more stable. So in order to be able to find a compound that's based on AC1903 but improves its properties, we really need to use the gold standard assay for ion channels, which is electrophysiology. But the traditional methods of electrophysiology are very laborious and time intensive. And so we wanted to create an automated assay that will ease this process. So just briefly here, what I'm showing you on the left is the traditional manual patch clamp method of electrophysiology, where you have a glass pipette that is pulled to a tapered end and then brought in close proximity to the cellular membrane, and at which point you will form a seal with the cellular membrane. But what we have at the Broad is this automated electrophysiology platform called the SynchroPatch that utilizes this 384 well plate format that has a glass bottom and has small holes drilled into the glass bottom in each well. So what this really allows us to do is to be able to record 384 cells in parallel, and greatly increases the throughput by which we can screen molecules for increased inhibition of TRPC5. So this is just a screenshot of what the instrument interface looks like. And what I'm showing you here is that I have been able to successfully develop an assay on this platform that looks at TRPC5 current. So what you see here in black is a baseline trace, so when the cells are unstimulated. But then we can add a known activator of TRPC5 called riluzole, and you see induction of these doubly rectifying currents that I described to you earlier. And importantly, we can then add compounds on top of riluzole and see whether or not they inhibit the generated TRPC5 current. And what we see for this specific compound is that indeed we do get inhibition. So here I'm just showing this in another way through what's called a diary plot, so tracking those current magnitudes over time. So this is baseline, and you see we add riluzole here and we get increases in magnitude, which is due to that doubly rectifying current. And then the same compound, when added upon on top of riluzole, leads to inhibition of the channel. And so what we can really do is now look at the percent inhibition, so comparing this epoch to this epoch, and in comparison to AC1903, be able to really start to ask, do we get specific modifications that will allow us to increase the efficacy of TRPC5 inhibition? So this is just to show you that with a subset of compounds that we recorded both manually and on the SynchroPatch, that there is good qualitative agreement between the two platforms, which gives us confidence in the results that we will get from the SynchroPatch. So here in this graph, what I'm showing you is just a subset of the compounds that I've tested using this assay. And on the x-axis here, we are simply expressing inhibition relative to AC1903. Which is shown by this dotted line. So what we're really interested in here is this tail of compounds on the right, which indicate compound efficacy, basically compounds that are much better at AC1903 at inhibiting TRPC5 current, and for some of these, as much as threefold better. And we've published some of these results already last year. So then what we can really do is we can take those compounds that look very promising at single doses, which was what the previous graph was showing, and test them over a range of concentrations to be able to construct what we call here as dose response curves, so that we can see over a range of different concentrations whether we really have improved the inhibition of TRPC5 current. And what we can see here is that a few of these compounds are left shifted with respect to AC1903, which is this heavy black marked line here. And what that means is that these compounds that are left shifted, they're able to inhibit TRPC5 at much lower concentrations. And so we think that this is very promising, and we're really starting to understand the modifications that we have to make to the AC1903 backbone in order to make it more effective at inhibiting TRPC5 current. So one of the remaining questions really in the field is whether or not the TRPC5 inhibition is relevant in humans. Because as you've seen from the previous slides, most of the work that's been done has been in rodent models or in heterologous expression systems. So we decided to really get at this question by leveraging new stem cell technologies. So Choa Kim and Mahesh, two talented scientists in the lab, took induced pluripotent stem cells, human-induced pluripotent stem cells, and derived podocytes from them using a podocyte differentiation protocol. So then I took these cells and I recorded from them using traditional manual patch clamp electrophysiology. And you see what that looks like in this image on the left. So this is my glass pipette, and I'm bringing it in proximity to this podocyte, which is in vitro resembles what we know podocytes look like. So they're these very large, flattened cells with these cytoplasmic extensions. And I can record current from these cells. So this is, again, just another current voltage curve, as I've shown you earlier. And then what I can do is add an activator of TRPC5, in this case, a compound known as englerin. And you can see this nice induction of that doubly rectifying current, and the shape, as I've described to you, gives us confidence that this is an ion channel of the identity that we care about, TRPC5. And to give us even further confidence in this, we can now add that TRPC5 inhibitor that I've described to you, AC1903, and indeed we do get inhibition of the englerin-induced current. So this now is, to our knowledge, the first time that TRPC5 channels have been shown to be functional in a human podocyte. And I just like to point out here that the early pharmaceutical work that generated AC1903 was invaluable to this research, because without the proper tools, then we wouldn't truly be able to dissect whether or not the currents that we're looking at are TRPC5 specific currents. So here I'm just showing you another current diary plot. So just tracking current magnitudes over time. You see this is baseline and this stem cell derived podocytes. You add englerin, we get that doubly rectifying current that is inhibited by application of AC1903. And when we stop application of AC1903, then we are able to restore those current magnitudes. And this is just statistical analysis looking at a population of these podocytes. So we are really confident now that human podocytes do actually express functional TRPC5 channels, which makes targeting TRPC5 a more viable therapeutic hypothesis. And this work has been submitted, and I won't have the time to go over this today, but we also show that AC1903 in this human-derived podocytes is able to prevent the damage caused by injurious stimuli. So what I've told you today is that we are able to screen through many different compounds to find inhibitors of TRPC5 current, which we know is protective against kidney disease. And we have proven that these TRPC5 channels are functionally expressed in human cells using the gold standard for ion channel work, which is manual electrophysiology. So with that, I'd like to thank the numerous mentors and collaborators by which none of this work would have been possible, in particular Anna Greca, who's the head of the Kidney Disease Initiative, our collaborators at the Stanley Center of the Broad Institute, and Corey Hopkins and his scientist Swagat Sharma at the University of Nebraska, who provided the compounds and have been great collaborators over the years. And with that, I'd like to thank you for listening today.
Thank you, Wanchi, for that very exciting work, including work that's not yet been published, very recent work. I'd like the audience to know that our first two speakers, Homer and Wanchi, have talks in other sessions in the 40th APAMS, and so does our next speaker. So I am to introduce Leodevico L. Vic Ilag. Vic is Chief Scientific Officer and co-founder of Xerion Limited. For the last 25 years, he has worked and co-founded biotech companies in Germany, Australia, and the Philippines. He has a PhD in microbiology immunology and did postdoctoral studies on structural biology of viruses. Vic?
Good morning, and thank you very much for the opportunity to present today. This is actually a broad topic to cover within 15 minutes. Therefore, I'd like to focus on the basics and the implications to COVID-19 infection and the associated cytokine storm. I would also like to discuss a way of measuring fatty acids as a biomarker for inflammation and state of wellness and its implications to the current pandemic. So I'd like to start with this slide to give an overview of the human defense system or human immune system. I've taken the figure from Miller et al. And essentially throws three major components. The first component being your physiological barriers, primarily the skin, which is composed of fatty acids, but as well as other factors, which are responsible for preventing or entry of any invading pathogen or such as a virus. However, if this physiological barrier is breached, you have the innate immune system, which is your second line of defense, and composed of immune cells as well as other factors. Now, while eliminating this pathogen, these cells are communicating with a next line of defense, which is the adaptive immune system, comprised of B cells, responsible for making antibodies, and T cells for cell-mediated immunity. So this adaptive immune system is specific and is actually the basis for obtaining long-term immunity from vaccines. So inflammation is an important biological process, especially in the acute form. So it's the body's way of getting rid of infections as well as repairing wounds. So if one has an infection or a cut or wound, this activates the innate immune system to recruit immune cells such as neutrophils to the site of infection or wound. This, once it arrives there, it secretes factors to increase the permeability to allow infiltration of more cells, but at the same time, leakage of liquid into the site of infection, generating an edema. So you notice if you have an infection or a wound, there would be swelling, and that is because of the edema produced. Now, once the pathogen or wound is eliminated, this leads to the resolution of inflammation, where infiltration is stopped because the vasculature is pretty much sealed and other remaining debris taken care of macrophages. However, there are instances wherein the vasculature is not completely sealed, leading to low grade of inflammation. If this low grade inflammation persists for long periods of time, that could lead to disease, and this low grade inflammation is often called chronic inflammation. So chronic inflammation has been attributed to a lot of diseases such as cancer, diabetes, depression, arthritis, etc., as shown in this slide. It's actually, as you will note, these are the same comorbidities which have been associated to patients who develop serious complications to COVID-19 infection. So Deepak Panigrahi et al. Recently proposed in an article, as depicted in this slide, that reduced levels of EETs and SPMs, epoxy-eicosaptrionic acids, and specialized pro-resolving mediators is responsible for generating a cytokine storm. So I'd like to walk you through this process. So once a virus infects a cell, this leads to cell lysis, producing cellular debris. The cellular debris then has to be mopped up by macrophages. Macrophages then produce eicosanoids, which then induce the production of cytokines. Cytokines actually then make the vasculature more permeable to allow entry of immune cells into the site of infection. And then this then goes through a cycle. Now, if there are more eicosanoids produced, that could lead to an eicosanoid storm, which then produces more cytokines and then producing more vascular leakage, leading to accumulation of fluids, which could lead to pneumonia. At the same time, it shuts down the immune system. Therefore, the authors proposed that this could be resolved by increasing the amount of EETs and SPM. So what are EETs and SPMs? They're actually metabolites of long-chain polyunsaturated fatty acids, omega-3 and omega-6, and I will describe them in the next slides. So DHA and EPA are the key omega-3 fatty acids with major biological roles. So the name omega-3 is based on the first double bond present on the third carbon atom. These omega-3 fatty acids, EPA and DHA, are synthesized by the body very inefficiently from a precursor, ALA, alpha-linolenic acid. That's why one needs to obtain it from dietary sources such as oily fish, sardines, salmon, and mackerel. The primary source of DHA and EPA are microalgae and seaweeds. So these omega-3 fatty acids are important structural components of all cells and have multiple roles. So it is generally associated with anti-inflammatory functions, but it also has antimicrobial properties. ALA is only a precursor and has less biological relevance. So in short, omega-3 fatty acids are anti-inflammatory. So the next group are the omega-6 fatty acids, and it's based on the name that the first double bond occurs on the omega-6 carbon. So the most important omega-6 is arachidonic acid, AA, which is produced from linoleic acid, LA. So linoleic acid is essential and found in vegetable, seed, and nut oils. So AA is the dominant form found in membranes and key mediator of inflammation and important in wound healing, growth, and infant development. So in general, omega-6 fatty acids are pro-inflammatory. So now this slide illustrates the biochemistry of inflammation. So if you have an inflammatory challenge, such as a cut, wound, or an infection, this releases arachidonic acid in the membrane by a phospholipase, which is then metabolized to a variety of pro-inflammatory eicosanoids. These eicosanoids are responsible for the production of more cytokines, which then triggers the whole downstream process of inflammation. If this is in excess, arachidonic acid, you would then have an excess of eicosanoids leading to eicosanoid storm and then subsequent cytokine storm. Now, this could be regulated by EPA and DHA. EPA and DHA form your EETs as well as SPMs, the resolvins and protectives, and their role is to resolve inflammation. They could actually work at different choke points as well. So EPA is actually a direct competitive inhibitor of AA because they share the same enzyme. So depending what the substrate it is, it could be pro-inflammatory or anti-inflammatory. At the same time, these anti-inflammatory eicosanoids produced from EPA could actually block the production of cytokines. So therefore, you could then see the relationship between omega-6 and 3 fatty acids. So in principle, one way to regulate inflammation is to have the right balance of AA and EPA and DHA. Unfortunately, there's no drug available to do this. However, this can be actually modulated simply through dietary intervention. So this slide exemplifies how diet can regulate inflammation. So as mentioned, omega-6 fatty acids are pro-inflammatory. So linoleic acid is actually converted all the way to arachidonic acid, but could be modulated by both DHA and EPA. As you'll see here, DHA actually inhibits the conversion of linoleic acid to the precursors prior to the production of arachidonic acid. EPA blocks the conversion of dihomogamma-linolenic acid, DGLA, to arachidonic acid, leading to the production or its conversion into anti-inflammatory eicosanoids. However, insulin promotes the conversion of DGLA and arachidonic acid. So if you eat a lot of carbohydrates, you produce a lot of insulin. And people with conditions such as type 2 diabetes, obesity, as well as hyperinsulinemia, such as PCOS, would then promote the production of arachidonic acid, leading to more pro-inflammatory eicosanoids. So how do we measure fatty acids? So the traditional way of measuring fatty acids in blood is via venipuncture, taking a needle, sticking into your arm, and extracting bloods. Red blood cells need to be isolated, and unfortunately, fatty acids are unstable in blood, even at low temperatures, such as minus 20 Celsius. The blood needs to be stored at really low temperatures at minus 180 Celsius to keep them stable. So analysis is by gas chromatography of derivatized fatty acid esters. This method is usually tedious and expensive and has limited its use. So fortunately, the problem of fatty acid stability was solved by Professor Bob Gibson and his then PhD student, Dr. Lou Gay, where they developed a dried blood spot card, which allows the stabilization of omega-3 fatty acids at room temperature for up to 9 weeks or 2 weeks at 40 degrees. It has high accuracy, where the results are very comparable between those obtained from dried blood spot as well as venipuncture. We actually licensed this technology. I have partnered with BASF so you could actually allow your own personal measurement of omega-3 fatty acids and other fatty acids. So one of the things you could do once you have your fatty acids is to measure your omega-3 index. So this is a metric developed by Professor Bill Harris and Professor Clemens von Schacke. As shown in this slide, it's the ratio of the omega-3 fatty acids, EPA and DHA, as a percentage of total fatty acids in red blood cells. So there are advantages of testing red blood cells, including its ease in sampling and that the EPA and DHA are stable in red blood cells for several weeks, providing a reliable proxy for the levels of DHA and EPA in all tissues and organs in the body. So what Harris and von Schacke actually was looking at, the reason they developed this method, they observed that populations with high omega-3 indices, those over 8%, have lower rates of cardiovascular disease, while those with low omega-3 indices, less than 4%, have higher incidence of cardiovascular disease, such that they recommended that the desirable range is between 8 to 12%. Based on a 15,000 male physician study, prospective study with matched controls, the omega-3 index was actually found to be a better predictor for sudden heart attacks compared to cholesterol and other biomarkers. So another ratio developed by Dr. Barry Sears is the AA-EPA ratio. As we know, EPA and AA compete for the same enzymes and determines whether pro or anti-inflammatory eicosanoids are produced. So looking at the intervention studies, Dr. Sears found out that with purified EPA-DHA, that the ratio of 1.5 to 3 will be optimal and is reflected in populations with low incidence of inflammatory disease, such as the Japanese and the Inuit from Greenland. However, those with higher ratios correlated with higher incidences of inflammatory disease, such as those in the West. So this slide is a bit of a tangent, but I think worth noting. So everybody has been talking a lot about the gut microbiome and its role in inflammation and the use of probiotics, but the mechanism of how this operates is not well understood. Nevertheless, there are studies showing a direct role of omega-3 fatty acids. It has been shown that EPA and DHA actually inhibit the formation of the NLRP3 inflammasome. So NLRP inflammasome is formed during gut dysbiosis and even actually after a viral infection. So, in another paper by Kalanan et al., they showed that using transgenic mice that constitutively produce omega-3 leads to the induction of intestinal alkaline phosphatase, which is an enzyme which inactivates the LPS or lipopolysaccharide molecule of gram-negative bacteria. So LPS is a key mediator of inflammatory reactions. It's actually the culprit as well in sepsis. So this slide is a good depiction of how coronary heart disease progresses, and actually its origins could be traced to inflammation, and actually inflammation plays a role throughout the process of forming of a blood clot. So it's tempting to speculate that by controlling inflammation early on, one can prevent progression of this disease. So blood clots, interestingly, have been observed in a lot of COVID-19 patients, and it's tempting to speculate whether this excessive inflammation leads to rapid development of blood clots. So I'd like to wrap up going back to the earlier slide I showed from Panigrai et al. And one could see that EETs and SPMs, which are derived from omega-3 fatty acids, can prevent multiple steps in a cytokine storm. First, it can boost the efficiency of macrophages from mopping up cellular debris without generating an eicosanoid storm. ETs, SPMs also block pro-inflammatory cytokines, as well as leakage into vasculature, preventing infiltration of immune cells. Moreover, it also activates the adaptive immune system, promoting the generation of antibodies as well as cell-mediated immunity. So although the authors propose the use of EETs and SPMs, one should be able to ask us, why not just increase omega-3 intake, since they are the precursors, through dietary intervention, such as eating more oily fish and seaweeds, or even supplements. It is a cheaper and safer approach that can be implemented immediately. So therefore, one could then look at optimal levels, or even propose that optimal levels of omega-3 and omega-6 fatty acids can prevent the cytokine storm once infected with COVID-19, especially in the event that a vaccine might not be available soon. So this is a testable hypothesis, especially with a simple and facile way of measuring fatty acids from a few drops of blood. So finally, I'd like to end up with this slide, my presentation, showing that there is actually a global deficit in omega-3 levels. So only 20% of the population meets the recommended EPA and DHA levels. It's tempting to point out also, and worth noticing, although without in-depth analysis, that countries in green with high levels of omega-3, such as Japan, Korea, and Norway, have relatively low COVID-19 death rates, while some of the countries in red with high death rates from COVID-19 have actually low omega-3 levels. Brazil, India, Italy, and the UK. So thank you very much for your kind attention. I'll be glad to entertain your questions later.
Thank you very much, Vic. We're also monitoring our participants on YouTube. There are a lot more people on YouTube than there are on Zoom. We are a little bit ahead of time, but that's okay, because we will have some speakers who have to leave after the morning Q&A. Our next speaker is Conrad Russell Cruz, who is developing cell-based immunotherapies for cancer and opportunistic infections. He expands and genetically modifies natural killer cells and T cells, which are the principal effector cells of innate and adaptive immunity, respectively, to specifically target malignant and infected cells. He received his medical degree from the University of the Philippines, Manila, and his PhD from the Baylor College of Medicine. He is currently an assistant professor of pediatrics at the Children's National Hospital, George Washington University in Washington, D.C., to speak on cell-based immunotherapy for cancer and HIV. Let us all welcome Conrad Russell Cruz.
Good morning. Thank you to the organizers of this conference, with special thanks to Drs. Concepcion, Pantua, and Hernandez for giving me this opportunity to share our work. Cell-based immunotherapies to treat cancer and HIV is not mine alone. I want to make sure there's enough time for this, so I'd like to acknowledge all the incredibly wonderful people behind these experiments and these stories. Here at Children's National, we established a cell therapy program called the Program for Cell Enhancement and Technologies for Immunotherapy, or CETI. One simple way to understand what we do, we work with the cells of the immune system, reprogram them, train them, make them more functional, using the tools available to us from the field of gene therapy, genetic modification, and genome editing, creating chimeras, and implement them in a true bench-to-bedside workflow using clinical trials that we sponsor. If you pull all these together, you have our logo, CETI, which not coincidentally is Latin for whale. Today's talk is meant to be a quick introduction, and I encourage you to read our papers or reach out to me to know more about these technologies. This presentation is divided into 3 short segments. I want to introduce you to cell-based therapies, and we'll be giving you 2 examples from 2 main platforms being developed by my group: natural killer cells, or NK cells, and T cells. Cell therapy is part of the immunotherapy revolution. Not as well known perhaps outside certain circles as their molecular counterparts, checkpoint blockers like nivolumab, for example, but getting increasing traction because of their continued promising results. Here you can see cell therapies featured in the non-scientific press, And featured over here is the literal poster child for T-cell therapy, Emily Whitehead, who after getting remission from her tumors using T-cell therapies, got to visit the White House and got an excuse letter from President Obama himself. Cell therapies involve the infusion of actual cells to treat cancer or infectious disease. As shown in this schematic, cells are obtained from various sources, modified in the laboratory by selection, expansion, and gene modification, and then reinfused back into the patient. Cell therapies are highly specific. They can recognize a diseased cell from a healthy one. In this video from a colleague, Dr. Nabil Ahmed, we can appreciate the specificity of these cells, homing to the tumor cell in the middle, ignoring all the other cells, and mediating clean program death of this cancer cell. What particularly makes us excited is that cell therapies can potentially confer lifelong therapy to disease, a consequence of immune memory. So here now are two examples of effector cells used for two very different diseases. The first example, natural killer cells, or NK cells, have been explored by our group and others as a platform to treat cancer. And I don't need to tell this audience that we have limited options for certain malignancies. Tumor heterogeneity is an important source of treatment failure. The tumor is able to escape therapies and therefore comes back with treatment-resistant disease. In this setting, natural killer cells are an attractive option. These innate immune cells, actively home to the disease site, readily lyse their targets without need for priming or knowledge of antigens as they recognize upregulated stress signals on diseased cells. They also have documented activity against several malignancies in vitro. Unfortunately, the tumor is a wily enemy. Besides its heterogeneity that can lead to immune escape, it also maintains a hostile tumor microenvironment. The tumor creates this environment by co-opting cells in its surroundings. This environment is unfavorable to immune cells. TGF-beta, an immune suppressive cytokine, has devastating effects on different immune cells like CD4 T cells, CD8s, including natural killer cells. We therefore devised a strategy to abrogate the negative effects of this cytokine by creating a dominant negative receptor. As you can see here, we used a retrovirus to encode a version of the TGF-beta receptor that does not signal. This dominant negative receptor effectively halts a signal that normally leads to phosphorylation of various intracellular signaling molecules that lead to production of immune suppressive genes. This is the general schema for how we make these cells. We first select the NK cell population from peripheral blood, expand them by stimulation with artificial antigen-presenting cells and cytokines, and subsequently modify them using a retrovirus to introduce our dominant negative receptor transgene. We have since published that we can get good transduction efficiencies of these cells, as shown here on the left, leading to cessation of signaling as shown by the lack of phosphorylation of SMAD2 in the presence of TGF-beta. These NK cells are able to maintain their killing in the face of TGF-beta enriched tumor conditioned media. In work by my student Alison Powell, shown here in blue, are normal cells that do show decrease in killing in the presence of the cytokine. However, in red are what we observe when we transduce these cells, and shown here that there is no drop in killing. The cells are unfazed by the cytokine and keep doing their function. In an in vivo model of neuroblastoma, performed by another grad student, Rachel Berga, we see that these cells are capable of conferring protection to animals engrafted with human tumors. As you can appreciate in this IVIS imaging of tumors that keep growing and lead to death in animals that are treated, untreated, or mock treated, and delay the tumor progression in animals given with our construct. This leads to better survival in animals administered with various permutations for dominant negative receptor construct in yellow here and in blue. To summarize, NK cells may be effective therapies for brain tumors and solid tumors, and they can protecting NK from immune suppressive cytokines can help them withstand a tumor microenvironment, and that we can do this using a dominant negative receptor. Our next example involves T cells for HIV. Our group's long-standing history with making T cells for different infections has allowed us to explore therapies for several different diseases. We can make T cells for a lot of antigens. We can make them for EBV and HPV and Mucor. And to all of these, we actually say, we have a whale for that. Here is a picture of the AIDS quilt in Washington, D.C. And it's important to note that DC has one of the highest rates of HIV in the country. DC has a prevalence of around 3.2%, which if you look at this comparison is actually higher than some of the African countries. making it an important source of morbidity and mortality in the region. There are several ways to use immunotherapy against HIV. T cells targeting infected cells, NK cells targeting virions on the surface, antibodies targeting free virus. We chose to combine all three in a single platform. We predict that creating synergy between innate antibody and adaptive immunity by endowing HIV-specific T cells with the ability to produce HIV antibodies designed to recruit endogenous immune cells can lead to long-term sterilizing cure. Here is a schema. T cells are generated from peripheral blood mononuclear cells and pulsed with antigen-presenting cells that are presenting overlapping peptide libraries against HIV antigens. These are then transduced to allow for expression of these antibodies. As you can see here, these HIV T cells can be modified with our transgene, transduction efficiency shown here, and made to secrete broadly neutralizing antibodies against HIV. These T cells maintain their specificity when you compare the untransduced from the transduced, and they keep on recognizing HIV antigens GAG, NEF, and POL. More importantly, the antibodies created by these T cells, present in the transduced and not in the untransduced, are able to bind to HIV envelope expressed by cells, as you could appreciate here. We then tested to see if these antibodies can neutralize free virus. We mixed these antibodies with virus and tested the ability of these viruses to infect new cells. The amount of neutralization is the amount of cells that have not been infected compared to untreated control. You can appreciate that there's a significant amount of the virus that is neutralized by these T cell secreted antibodies. We also tested the antibodies' ability to recruit NK cells to mediate antibody-mediated antibody-dependent cell cytotoxicity, or ADCC. As you can see, when NK cell killing is increased in the presence of antibody, similar to what can be seen in controlled non-T cell secreted antibodies. Finally, we looked at the synergy between these cells in a virus inhibition assay. As you can see here, after we infect T cells, co-culture with HIV and specific T cells lead to a decrease in virus production, showing efficacy from the T cell arm, a further decrease when these are transduced, showing efficacy from the antibody, and still a further decrease when co-cultured with NK cells, showing the effects of ADCC. To summarize, the T cells can be modified to secrete engineered broadly neutralizing antibodies, and they can mediate a tripartite response to HIV. In closing, I hope I have convinced you that cell therapies are promising new therapies. They are safe, they potentially provide precise therapy, they can be expanded to clinically relevant numbers, they can be modified to confer additional functionality. There are lots of future directions, and we refer you to these papers to talk about this. But we know we have a difficult task ahead of us, but a very exciting one. After all, as Carl Jung says, if the path before you is clear, you're probably on someone else's. I hope there are a fair number of students and trainees watching this conference, and to them, I close with something I tell my own students. I hope that attendees of this conference will someday resemble the attendees of another conference organized almost 100 years ago. The 1927 conference attendees would subsequently feature several Nobel laureates and scientific luminaries. I dare you all to do better. Thank you for listening. I'd like to acknowledge the sources for our funding here and my very hardworking laboratory, both past and present. And of course, special thanks to the leadership of my program and department, and particularly to my mentor, Dr. Kath Pollard. If you have any questions or interested in collaborating, my email address is up here. Thank you so much.
Phew. Thank you, Russell, for that fantastic talk. So you shared with us how you're making the promise of immunotherapy closer to reality. Okay, so thank you for joining us at short notice. Russell came in as another speaker in this session quite late, and we were already quite oversubscribed in the session, but I'm so glad you made it, Russell. So now, ladies and gentlemen, the floor is open for your questions. And so we have our 4 speakers in the basic research field, in the molecular and cellular sciences, ready to take your questions. So question for Homer: what is the antibiotic class of globomycin? if it becomes a therapy option, is it for human and animal use? And what are the risks and chances of resistance formation? Homer? Oh, wait. No, unmute you first. Unmute you first, Homer. Repeat.
Yeah, so globomycin is a natural product. It's a cyclic molecule. If it becomes a therapy option, we are hoping for humans, but right now we are pretty far from going to the clinic because we're experiencing preclinical hurdles right now that I can't actually tell you. But it's still far. What are the chances of resistance formation once globomycin is used? So the resistance formation, the frequency of resistance with globomycin is very low. So there are 3 ways that it can actually resist, the bacteria can resist globomycin. But as I've told you, even though there are resistance, they are still sensitive to serum and they are attenuated in vivo. So it will not, most probably it will not persist given the frequency, the very low frequency. But it's still far. It's still far.
Okay, thank you, Homer. Are there other questions? So, Richmond, do you see any other questions?
None, ma'am.
Okay, maybe we can wait a while for people to ask questions. So first I have a question for Russell. So Russell, how much further down the line do you need to get a T cell expressing an antibody, closer to proof of concept and then to preclinical? So at what stage do you think you are in?
Yeah, so for the on the HIV side, so we're actually doing it for both the cancer side, which I didn't present today, and the HIV. On the HIV side, we are doing the mouse models with NSG mice, engrafted with human infected human T cells, and so hopefully after we've demonstrated proof of concept there, we could proceed with clinical trials if that's they show promising results. The good thing about our platform is so the HIV specific T cells are actually on clinical trials right now, and they're being given. We're not stopping art or anything, right? So it's still a safety trial, but those are non-gene modified ones. So after we demonstrate efficacy in the mouse model, then we'll proceed with hopefully giving them to patients soon.
There is a question for Vic Ilag from Anna Serkina. Okay. Are there any current studies on testing the effect of omega-3 on COVID-related inflammation? So Vic, the question.
Sorry. I was had to unmute. My recollection, there are 2. One thing I could remember, I think, is KD Pharma in, I think, Denmark, Norwegian, or Norway. The tricky thing, though, with, I think, treating omega-3 late to the game is that you could see that it's upstream. So if you have already inflammation going through, or let's say the vasculature is already severely leaked, it might be a bit too late. But I think it's still worth trying, but just something of a precaution. So I'm thinking if you want to do it, the analogy I usually make for inflammation is like a leaky roof. So your vasculature is leaky, so you have a leaky ceiling. And if it's dripping on your furniture, let's say you don't really notice it. You're damp, but once it gets damaged and that ceiling collapses, it's a bit difficult to seal the ceiling. So, but very good question, yeah.
Okay, thank you, Vic. Another question to Homer from Francis Prel III. Dr. Pantua, can you comment on the pharmacokinetic profile of G0790? Is it orally available?
So just to answer that, so I just want to give you a brief history of this molecule. So G0790 is the first compound that we made from globomycin. So it's not the compound that we are actually pursuing for further preclinical studies, but the PK/PD of G0790 is fairly okay, but G0790 still has the ester linkage, so it's still sensitive to esterases in the serum, and if I can share with you, we has just published the SAR, the structure activity relationship paper in the bioorganic medicinal chemistry of more potent and better PK molecules version of the globomycin, but it's not 0790. In terms of the oral availability, we haven't tested any. We haven't done any oral administration of this compound, even with the highly potent globomycin analog. So we don't know.
Okay, thank you.
I have a question for Dr. Cruz from Jenny Baban on the YouTube chat. Current immune cell therapies are autologous using the patient's own cells. Do you have any comment on allotherapies using a universal cell bank to generate designer T cells? Russell?
Thanks, Jenny, for your question. So right now, our clinical trials are actually a mix of autologous and allogeneic therapies. Some of our virus trials are more advanced, and those are actually using cell banks. So these are in the setting of transplant and for primary immune deficiencies. And these are being led by another PI in the laboratory, Dr. Mike Keller. And we find that these are actually, pretty safe, because after we've cultured these cells for a while, you basically weed out the GVHD causing allogeneic cell products, and you're left with those that are recognizing your antigen of choice. So we hope to develop, this cell bank further. One thing I think we have an advantage of over other platforms is, the generation process, not only makes the T cells more specific, like I mentioned earlier, it also removes the alloreactive components. And so, yes, that is the plan for both the tumors down the road. Currently, it's only for EBV, CMV, adeno, and other viruses post-transplant and in immune deficiencies.
Thank you. So I have a question for Juan Chi. So Juan Chi, your compound, what is its source? And then how are the analogs decorated in general? And so did you try to retrofit this with a TRPC5 model? So is the structure of TRPC5 well resolved, considering its membrane? It's a channel. So Juan Chi?
Yeah, thanks for the questions. Those are all great questions. So the original molecule AC1903 is a benzidimidazole-based compound. So it's a synthetic compound, and we found several modifications that improve it, mostly dealing with the substituent at the R1 position. So we found you can change those groups, and certain groups make it much more potent than the original molecule. There is a TRPC5 structure, actually very recently published a few months ago, and it seems very likely that the binding site for AC1903 based on that structure is in a region of the molecule that we've long known stabilizes the pore. And so that paper did not crystallize, or rather it was a cryo-EM paper, so it did not have AC1903 in the complex. But based on the lipid binding sites, it's very likely that AC93 is in this site that stabilizes the pore, and so that's probably how it's blocking the channel.
So there's a question for Vic Ilag from Al Sarafica. Are we able to produce omega-3 from seaweed in the Philippines?
Well, I think the primary source right now of omega-3 is actually sardines. So actually also from fish waste, they extract it. And that's why Norway is the number one producer, I think, from salmon, from the processing of the other components, so they could do that. So in seaweed, yeah, technically it should be. I think in Australia, actually, there are a lot of ponds which are used to grow microalgae. And I think, if I'm not mistaken, I think the Philippines has a spirulina program or seaweed or microalgae program. Actually, that's that's sources used for producing omega-3. And actually, there, although I think the focus is biofuels, I think we've done this already as well, our calculations. It's just not economical to make biofuels from seaweeds. You're better off to extract probably omega-3s. So something you might want to consider. But the extraction, I think, should be pretty much more efficient. And then you want also to separate like EPA and DHA specifically, because you feel usually when you get your omega-3s from fish, it's a mixture of DHA and EPA. But the market now is to go for DHA as well as EPA. And the advantage also of algae or seaweeds is for the vegan population, because they don't like to have fish. So that would be another market. And yeah, it's a high value product. So if you're, yeah, so just to reiterate again, yep, I would rather go, rather than focusing on biofuels, you probably want to go to the high value omega-3 fatty acids, DHA and EPA.
So, Vic,
Thank you.
I share that with my friends, and they said, "Oh, we love sardines, but then we have a high uric acid." So,
Yeah.
is
That's
that
just like cholesterol. I think it's a misconception. I don't think you can really get uric acid. You probably get more uric acid. You produce it when it becomes a problem rather than from dietary
okay?
purpose.
So we'll follow that lead. So there are more questions from Anna Serkina to Russell Cruz. Will quasi species of HIV-1 be a hurdle for your immunotherapy?
Yeah, thank you for your question, Anna. So the short answer is yes. It's potentially an issue for the immunotherapy. I guess the beauty of the platform is it relies on a curated set of peptides, that you could custom, to whatever the antigen is in your patient sample. The other thing that we've also found is, and this is in collaboration, my mentor's collaboration with Brad Jones, who's an HIV leading expert in HIV, is there are these escape mutants that seem to predominate after ART. And by targeting those, we hopefully, because we're giving these therapies, at least in the beginning, after ART or during ART, then these cells could potentially go after those. And again, like we sort of tried to put together, like a platform that targets different components of the immune system. So even if some do escape the T cell arm, they'll be addressed by the NK cell arm or the antibody arm. And so hopefully by engaging all multiple components of the immune system, you can avoid some of these escapes.
Okay, so from Ronilo Flores to Dr. Cruz, again, in your experience, how important is or crucial are the unique post-translational signatures, specifically glycosylation, as targets for immunotherapy?
Yeah, thank you for your question, Renilo. So I guess very, very roughly, like there are two ways that T cell therapies are being used right now to target cancer and I guess in some opportunistic diseases, right? Like one is like straightforward genetic modification using chimeric antigen receptors. And I think this is what you're sort of referring to. So they target intact proteins on the surface of these diseased cells, right? And so in that case, things like glycosylation or other post-translational modifications do matter. The way we use them is we rely on the endogenous T cell receptors for these cells. And so they actually target, processed peptides. So, and most of them would be internal, right? And so these are chopped up and what's important is the sequence. So when they go up to the membrane, shuttled by MHC, their modifications won't matter. So for the CAR T cells, yes, it will. And for the TCR-based ones, and not so much.
Okay, thank you. I have another question for Juan because I'm so interested in his work. Can you tell us a bit more about that new instrument that you are using that is able to do multi-screening? And First of all, what kind of cells do you use? We just use the oocytes, the frog oocytes for electrophysiology. In your case, what kind of cells do you use? And then you're able to do this on a 384 cell plate.
So it's quite adaptable. You can use heterologous expression systems. Those are the most common because you need to be able to scale up, right? That's the biggest hurdle probably, is being able to get large quantities, millions of good quality cells, because in electrophysiology, the integrity of the cell membrane is the primary hurdle. So to be able to grow up such a big batch of cells, you usually need heterologous expression systems, but we have been able to patch on induced pluripotent stem cells that have been differentiated into, say, cardiomyocytes, other electrical excitable cells. You can do neurons, primary culture. So it's quite an adaptable platform, but your cells have to be okay with being in suspension because they have to sink to the bottom of that well, and you have to be able to produce quite a lot of them.
Yeah, that's something that we should continue to talk about much. So, do we have time? So, Joey, how are we on time? Are we okay? Yes. So, are there any more questions? If not, we will proceed with the next equally interesting part of our session. Any more questions? I'd like to thank again our speakers, our first four speakers, for such mind-stimulating talks. And at this point, I know that some of them have to leave. So Vic Ilag and Homer Pantua are joining a meeting of the DOST-PCHRD. And I think, I don't know, Russell, are you here to stay for a while?
Yeah, I can stay for a while, yeah.
Fantastic. And Juanchi as well? Okay, so now we'll move on to...
Thank you very much.
Thank you all.
Thank you, everyone.
So I'd like to move now to a very important part of our session. And this is the talks of our medical doctors based in UP Manila and the Philippine General Hospital. So Edward HM Wang, MD, Ewang for short, we call him Ewang, is professor of orthopedics at UPM, where he is also a university scientist. And I think he's known as an excellent professor to many of the younger MD students and graduates. He is program leader for SIBOL, the biomedical program, which he will talk to us about. Aside from SIBOL, his team recently set up the Office of Research Integrity for UP Manila. He is an academician at the National Academy of Science and Technology. Dr. Ewang? Yes.
Thank you. Good morning, ladies and gentlemen, and good morning to our session hosts, Professors Concepcion and Lapeña. Thank you for inviting me to present at this convention. I've chosen to present to you a UP Manila College of Medicine program called SIBOL. It stands for Surgical Innovation and Biotechnology Laboratory and has for its objective the innovation, creation, and manufacture of medical and surgical devices using locally sourced materials. The primary objective of which is to address the inadequacy of locally made healthcare equipment and devices. I have nothing to disclose in relation to this presentation. Neither do I have any business interests associated with any of the SIBOL projects, except for my involvement as primary investigator in one of the projects. This is a simple outline of my talk. It is not so much a presentation of a research question, but more a description of our journey to set up a biomedical program at the UP Manila. I will then show you how we responded to COVID, and finally, I will share with you reflections and lessons we continue to learn about such a program. Let me start off with this slide, which rings and resonates every single day of our professional lives. Almost all medical devices present in developing countries have been designed for use in industrialized countries. Consequently, they are often unaffordable and are maladapted to low-resource settings. Take a look at all of these different health devices and technologies, even those sorely needed during this pandemic, including ventilators and PPE. Everything is imported, Is it any wonder why we always do not have enough? Why we always cannot afford them? And worse, why the spare parts are never available? So why don't we make them? Yes, we can, but we need the engineers and the clinicians to work together to design the devices. However, while we do have individual clinicians and engineers doing this, there are to date few such mature institutional linkages between clinicians and engineers, whether in government or the private sector. This lack of collaboration between engineers, scientists, and clinicians is a situation we also see at the University of the Philippines, where two great masses of brain cells, one in Diliman, the engineers and scientists, and one in Manila, the clinicians and allied medical staff, hardly ever meet, seemingly separated by the deep divide of the Great Pasig River. How many times, after being frustrated by the unavailability of some medical equipment in the hospital, have healthcare workers in the UP Manila thought about or actually assembled a rudimentary backyard solution, only to shelve it because of the lack of an integral engineering component or the lack of its understanding? Engineers in UP Diliman, on the other hand, awakened to some of the greatest eureka solutions to health problems, only to find out the healthcare provider does not need that earth-shaking creation after all. Sometime in March of 2019, then UP Diliman TTBDO, or Technology Transfer and Business Development Office Director Michay Pasha, sent to the UP Manila a list of engineering projects requiring clinical trials. Together with NIH Executive Director Eva Cotionco de la Paz, we set up what we called a CLIck meeting, conversations leading to innovations and collaborations. This marked the start of the construction of a bridge across the Pasig. We had a teleconference, the UP Diliman group on one side and the UP Manila group on the other side. The meeting was a success. We matched some of the engineers and scientists from the UP Diliman side to the clinicians on the UP Manila side, so they could continue from conversations into collaborations. And then we decided to cross the bridge to UP Diliman, finding out in the process that we didn't, after all, need a visa to enter that republic. We made a presentation entitled Getting to Know Each Other at the meeting sponsored by the UP Diliman TTBDO at the College of Engineering to a large group of engineer and scientist researchers. This was a list of topics we brought to the audience. Let me show you a problem which received a lot of question and attention. The upper right-hand picture shows you a common injury at the PGH, where the skin and soft tissues of the leg are completely ripped off in a vehicular accident. A good method to cover this defect is using what we call vacuum-assisted closure, or VAC, VAC for short. There are now many different VAC apparatuses which are portable and convenient for patient use. These commercially available devices are imported and are unaffordable for the PGH patient. While they seem simple to make, we clinicians don't know how to. And the best we can do is shown on the right lower picture. The entire construct on the leg connected to a vacuum provided by a wall suction, which is why in the PGH it is more appropriately called wall suction assisted closure, or WASAC. The engineers must have taken pity on this WASAC method of closure because this project received endless questions and suggestions. It was a good meeting, and we took it one step further. We now arrange for the UP Diliman engineers and scientists to cross the bridge and visit the PGH, so they could see for themselves the problems they were creating solutions for. So 2 months after the initial click, we had visitors from UP Diliman at the hospital. These are pictures showing the chair of the operating room welcoming them, and the bottom pictures show how the engineers studied equipment and how they visited patients who were hooked up to the now infamous WASAC. As a result of this encounter, 4 projects were undertaken, all collaborations between UP Manila clinicians and UP Diliman engineers and scientists. We submitted these projects to the PCHRD together with a project for the establishment of an office and laboratory for research and development. In the process, the program was christened SIBOL, a Tagalog term meaning germination, and which appropriately stood for Surgical Innovation and Biotechnology Laboratory. By the end of the year, we presented the program to the Execom of the OST and were gifted for Christmas with a grant for SIBOL. But alas, no good run lasts for long. Just before the signing of the MOA between the DOST and the UP Manila, we were overtaken by COVID-19. And things happened in quick succession, and before we knew it, we were on complete lockdown. By the end of the month, what started as the Ides of March had seen an endless litany of friends and colleague frontliners whose lives had ended too soon. These personal tragedies, together with the spirit of bayanihan, made SIBOL ask what it could do in the face of this viral onslaught. We had no MOA, we had no money, we had no infrastructure, we had no equipment. Hey, but hold on, we had a reason for being. And most importantly, we had Viber and we had Zoom. So SIBOL convened March 20, and 3 months later, on June 20th, we had 11 ongoing projects. We divided our thrusts into 3: PPE, disinfection, and telemonitoring, basically to protect, to disinfect, and to distance. Here you see the 3 teams and 11 projects with the principal and co-investigators, always 1 clinician and 1 engineer or scientist. Under the PPE team, we have the reusable mask, the PAPR, which stands for Powered Air Purifying Respirator, and the coveralls. Under the disinfection team, we have the Sanipod, the negative pressure room, clean intubate, PPE disinfection, and AeroSafe. And under the telemonitoring team, we have the telepresence, telemetry, and e-step. I'm unable to show as much as I would like to because of non-disclosure agreements. Here on the left upper picture, you see the Zoom meeting for the PAPR group. The left lower picture shows you products of the telemonitoring group. The right upper picture shows you the efficacy testing for the Sanipod, a sanitation chamber for healthcare workers in full PPEs. And the right lower picture is that of the group working on the e-stethoscope. Most of these projects have applied to the DOST PCHRD for funding. But while funds have not yet arrived, prototypes and expenditures have been shouldered by private donations. In this last part of my talk, I will share with you the little experience we have had over the past 1.5 years, getting SIBOL going and handling the COVID task force. What has SIBOL achieved in its infancy year? We have been able to initiate conversations and collaborations. And these collaborations have been both horizontal and vertical. Through work and by word of mouth, SIBOL has begun to draw interest from both students and faculty. Importantly, we have highlighted a multidisciplinary team approach. And in a short span of time, we have been able to produce several prototypes. What strengths have we discovered in the process? Like everyone has always said, the expertise and the capability are there. We have available mentors, especially those who have been successful abroad and who now share their knowledge and experiences with us. Individually, some of us have access to different support networks. And finally, not enough can be said about the robust existing research infrastructure in the university. We recognize our weaknesses too, and I have classified our weaknesses into those within our institution and those outside of our institution. Many of us have a poor comprehension of the process of translational research. Available mentors are a strength, but because there are not many who have the experience we look for, it can also be a weakness. Linkage with industry is strong, is starting, but is not yet robust. The Manila and Diliman campuses have no previous official collaborations on similar programs, which can serve as a template for us. Outside of the institution, we find there's a lack of a roadmap for requirements and compliance regulations for different technologies we want to develop. There's similarly no ready list of government agencies or offices in charge of standards for procedures and products. Finally, we were extremely handicapped by not being able to procure materials for our prototypes or access testing machines during lockdown. As part of the weaknesses, we need to talk about finances. From initiation of meetings to finally receiving MOA and budget, the fastest it took for some of the projects was 3 to 4 months. We commend the DOST for expediting the process, and we are grateful. However, a raging pandemic waits for no one. And if it were not for our private donors, we would never have been able to come up with our prototypes. As the cliche goes, there is always opportunity in crisis. We are seeing the opportunity to get different experts together from both engineering and science departments on the one hand, and medical and allied medical departments on the other, all with a single goal of fighting COVID. But we cannot rely on passion alone for long. We need to institutionalize this collaboration. And to make things much easier for future projects, we need to institutionalize the process and print out the roadmaps. What threats do we perceive pervade the innovation environment? We fear the continued difficulty in accessing necessary materials and equipment during quarantine. We are well aware of bureaucratic delays in government procedures, despite the officials doing their best to expedite matters. Because of the difficulty of the entire process, some of the researchers may very well give up soon after the prototype is made, especially when they see how much faster prototypes and final products are deployed by private institutions and industry, even if some of these do not really meet standards. From our experience, we can see that innovation processes such as these require more government support. I end my ruminations on a high note, but also on a very cautious note, because our curve continues to rise. There are still a myriad COVID problems needing solutions. And even when COVID-19 has waned, the problems and inadequacies of Philippine healthcare will continue, and they can overwhelm us. SIBOL senses the coming storm, and it has taken root and has begun the process of germination. Allow me to acknowledge these institutions and individuals, and most of all, the extreme efforts of all the members of our SIBOL teams. Thank you very much for listening.
Thank you very much, Ewang, for that wonderful presentation. We will entertain questions later. Our next speaker is Dr. Marie Carmela Lapitan, a practicing urologist at the Philippine General Hospital. She is currently a research professor of the University of the Philippines, Manila, National Institutes of Health, as well as its current Deputy Executive Director. She is a clinical professor and assistant chair for research of the Department of Surgery of the UP College of Medicine. Here to speak on the UP-PGH Department of Surgery Surgical Site Infection Surveillance Program, let us welcome Dr. Mela Lapitan.
Good day, everyone. It is with great pride and honor that in behalf of the Department of Surgery of the UP College of Medicine and the Philippine General Hospital, share with you our surgical site infection surveillance program. It is our department's focal quality improvement effort and also one of our core resources of our clinical research. It is the story of how surgeons, as a group of clinician scientists, when faced with patients whose wounds weep, reflect and take action on the consequences of what the scalpel in our hands did. The Department of Surgery Surgical Site Infection Surveillance Program started when we joined the Global Surgery Collaboration, a worldwide research community of surgeons, in its Global Surge 2 study in 2016. It was a study that collected and analyzed information about gastrointestinal surgeries and their surgical site infection rates. To do this, we started a surgical site infection surveillance program to ensure that the 30-day follow-up required by the study will be done. Realizing the feasibility and, more importantly, the valuable information that we are able to collect in implementing such a program, we extended the program beyond the Global Surge 2 data collection period and included breast surgeries in the surveillance. With a research grant from J&J in 2017, it allowed for the hiring of an SSI nurse to do active surveillance for 10 types of operations for year. From 2018 until the present time, the program is funded by the Foundation for the Advancement of Surgical Education and the PGH Expanded Hospital Research Office. In March this year, unfortunately, due to the COVID-related restriction of surgical services, the program was suspended. However, just last week, we have since restarted the program. The program has several components, including the institution of a department's policy requiring the practice of surveillance for surgical site infection for all our operations, a patient education package for SSI in monitoring and reporting, an active SSI surveillance by a dedicated team for selected patients, a reporting system for SSIs, and a practice audit research program on SSI rates and prevention. In August 2016, the Department of Surgery adopted a policy on surgical site infection surveillance that required proactive monitoring for surgical site infection for all our surgeries with a diagnosis of SSI following the CDC criteria. Surgeons were made to document the status of the surgical wounds at the time of discharge and at 30 days follow-up post-op. For selected patients, the SSI nurse would also follow up these patients and inform the respective surgeons if SSI is detected. The SSI team composed of 4 people filling 2.5 full-time equivalent roles. A part-time research assistant is assigned to identify the cases who will be actively monitored by the team. He also prepares the surveillance forms and collects and encodes the data in the SSI surveillance registry database. Another part-time secretary is assigned to call patients to remind them regarding their 30-day follow-up. She also assists in preparing the SSI reports. The SSI nurse, the only full-time staff in the team, engages with patients to educate them on wound care, surgical site infection monitoring and reporting, and advises them on their follow-up. Our administrative research assistant in the department oversees the preparation and dissemination of the SSI reports from the data collected and collated by the team. Listed here are the operations monitored by this team in 2019. And here are the cases monitored in 2020. An SSI surveillance report is prepared by the team on a bi-monthly basis. This report, particularly the SSI rates for the index operations, is incorporated and discussed in the pre-op post-op conference of the different divisions. The reports include the SSI rates for the different index operations being monitored, the SSI rate per surgeon per operation, and the different SSI surveillance issues and problems encountered during the monitoring period. Here is an example of a report on the SSI rate for simple open appendectomy for April 2019, as performed by the different divisions. Here is an example of a report of the SSI rate of one surgeon for the different operations he has performed in 2018. This report is forwarded to the Residency Training Committee and is considered in the clinical competency evaluation of the resident. Now here are our SSI rates in 2019 and in the first quarter of 2020. In the last column of the table are the published global rates for each operation for comparison. As you can see, we have a serious problem with our simple open appendectomies and more so with our laparoscopic appendectomies when it comes to the surgical site infection rates. But our rates for mastectomies and colorectal surgeries seem to be within the usual rates. Our SSI rates for our gallbladder and pancreatic surgeries and for our closure of stoma are at par with the global rates. The program gave us valuable insights of our patients' uncertainties and concerns about their surgical wounds. Also, Our patients' feedback provided us the motivation for us to continue with our work. Beyond the global Surge 2 research and the various publications from the Global Surgery Collaboration, the SSI surveillance program provided a rich source of data for the generation of many research papers for our consultants and resident staff, works which were presented as well as published in the different scientific forums. As a quality improvement effort, the SSI surveillance program triggered the development and implementation of 2 more programs in the department in the effort to prevent or lower our surgical site infection rates. The surgical antibiotic use guidelines in 2019, which also addresses the serious problem of antimicrobial resistance, and the surgical wound care standard in 2020. These programs are positioned to generate more scientific researches in our department. Thank you for your time and attention in listening to how UP-PGH Department of Surgery strives to turn our patients and our own wounds into wisdom. Good day.
Thank you, Dr. Mela, for that very, very serious concern in the clinics in the OR. And we have, each of us, I think, has some direct experience with relatives and loved ones who have experienced infections after surgery in PGH. So at this point, we'll move to our last and probably one of our most important topics today in biomedical research and other areas of research. And this is the subject of the talk of Dr. Jose Florencio Lapeña, MD, my co-host. And to introduce Joey, he is a professor of otorhinolaryngology at UP Manila, where he is also a university scientist and professorial chair holder in pediatric otorhinolaryngology, craniomaxillofacial surgery. Craniomaxillofacial surgery. He received the Publins UK inaugural Certified Sentinel of Science Award 2016 for, quote, handling the highest number of manuscripts across all the world's journals, unquote, among editors, earned the title of Star Reviewer Emeritus of the American Academy of Otolaryngology, Head and Neck Surgery Foundation, and won the 6th Eric Nubla Excellence Award as overall outstanding professional among all professions and most outstanding professional of the year in medicine 2017 from the Professional Regulation Commission of the Republic of the Philippines. Dr. Joey Lapeña.
Good morning. I will speak on authorship controversies, gift, guest, and ghost authorship. I am Secretary of WAMI, past President of APAMI, Charter President of PAMG, and Chair of the National Journal Selection Committee for the Western Pacific Region Index Medicus. Apart from these, I have no conflicts of interest to disclose. The reference for this lecture is available online at this site. Authorship controversies are often committed but seldom discussed. In this session, we will discuss principles of authorship and authorship criteria, gift, guest, and ghost authorship, and the order of authorship and authorship controversies. There are two general principles underlying authorship. First, all persons designated as authors should qualify for authorship. This means that everyone whose name is listed as an author should meet all the authorship criteria. Second, all those who qualify as authors should be so listed. Oftentimes, a postgrad student or resident in training who is no longer connected with an institution when the final manuscript is submitted for publication is left out of the list of authors because he or she is no longer around. That is inappropriate. Each author should have participated sufficiently in the work to take public responsibility for its contents. The recommendations that are made and updated annually by the ICMJE, or International Committee of Medical Journal Editors, has very specific sections on authorship concerns. Credit for authorship should be based on these criteria established by the ICMJE. First, all authors should have made substantial contributions to the conception and design or acquisition of data or analysis or interpretation of data. If there are three of us working on a research and one of us conceived the study and designed it, and the second person acquired the data, and the third person analyzed and interpreted the data, do all three of us meet the first criterion? Yes, we do. But there are three other criteria that we all have to meet. The second criterion is that all authors should be involved in drafting the article or revising it critically for important intellectual content. This means that editing a work or copy editing or proofreading it alone do not suffice for meeting the second criterion. If as an editor I help authors improve the quality of their paper by editing language, this does not mean that I meet the criteria for important intellectual content. Third, all authors should give final approval for the version to be published. Often, a journal will provide galley proofs of the final copy before publication, and it is this copy that all authors have to approve. This will prevent an author from making complaints in the future or saying that he or she does not agree with a certain part of the manuscript. And finally, and the most recent addition to the four criteria, all authors must agree to be accountable for all aspects of the work as far as any accusations of misconduct may be concerned. And this will prevent authors from washing their hands and finger pointing in case there is misconduct that is later on discovered by others. So, authors must meet all four criteria, and these are typically verified in writing in the manuscript submission process. Authorship responsibilities. All those named in a list of authors should live up to all these responsibilities. Honorific or gift authorship is unethical if only acknowledgement is due. Because all listed authors are liable for the published work, anonymous or ghost authors cannot be held liable, and therefore this is also improper. Bestowing honorific authorship on a superior, such as a chair, head of department, or director, is called gift authorship. It is usually practiced for political purposes, such as future promotion, and is usually involves the knowledge and consent of the superior. The superior, however, does not meet authorship criteria. And therefore, you have to be aware of how pronouns such as we or I are used in the manuscript as well as in the acknowledgements. Often, the manuscript will use the first person I, and suddenly the acknowledgements will say, I thank my, in this case, co-author of the paper. And you cannot be both an author as well as an acknowledged person. Deceit and fraud in medical research are a serious problem that we must all be concerned with. The most famous case in Britain is perhaps that of Malcolm Pierce, a senior lecturer whose head of department, Geoffrey Chamberlain, was professor when they both published a paper in the British Journal of Obstetrics and Gynecology. Incidentally, Pierce was assistant editor of that journal, where Chamberlain was editor. The paper claimed success in reimplanting an ectopic pregnancy, and in the same issue, Pierce had published a randomized controlled trial. A few months later, a whistleblower claimed that the reimplantation case was fictional, and so were the patients in the randomized trial. This scandal led to Chamberlain's resigning and Pierce's being struck off the medical register. Guest authorship refers to including an influential or well-known person among the list of authors. This is often done to boost the prestige of the manuscript or increase its chances of favorable review and publication. Again, it is usually with the knowledge and consent of the guest author because it is considered a win-win situation. The famous guest usually does not meet authorship criteria. The scandalous recent case of bogusly cloned human embryos claimed by Hwang Woo-suk of Korea and his longtime partner, Gerald Chatton, rocked the scientific world. Unfortunately, the panel that the University of Pittsburgh convened to investigate Chatton cleared him of misconduct and concluded that all he did was misbehave. Whether you agree with this or not, the fact of the matter is that these two had a long history of collaboration, including claiming to clone the world's first dog, here in the picture. Ghost authorship refers to professional paid writers who anonymously produce material that is then officially attributed to another author. This is often practiced in the pharmaceutical industry, where ghosts write promotional and favorable studies for drugs, and well-known persons such as professors, scientists, or senior clinicians are subsequently listed as authors. Often this is done with their consent and with more than adequate compensation. Very famous examples include Redox, which actually paid Richard Atkinson $1,500 in exchange for putting his name on the finished piece. He was so happy with the ghostwriter's work that he wrote, "Let me congratulate you and your writer on an excellent and thorough review of literature, clearly written. Perhaps I can get you to write all my papers for me." A year later, the drug was pulled from the market as doctors began reporting heart valve injuries in as many as 1/3 of patients taking the drug. There are many other drugs that were in a similar situation. A medical education company actually wrote articles supporting Neurontin for Park Davis, and $1,000 each was paid to friendly physicians and pharmacists to agree to author these articles. Zoloft documents had been written
that included 81 different articles promoting its usefulness for everything from panic order to pedophilia. And for some of the articles, the name of the author had still to be determined, or was still to be determined. Vioxx was a deadly drug eventually blamed for over 60,000 deaths, but it was also linked to shameful scandals relating to ghostwriters to boost sales. It involved the New England Journal of Medicine as well as the Annals of Internal Medicine. Order of authorship is a source of unhappiness and dispute. Not all co-authors do their fair share of the work. Bibliographic listings name only the first 1 to 3 or 6 authors depending on citation format. This dilutes the value of publication for those who aren't listed first. Moreover, scholars with seniority may insist on listing their names first, even though more junior scholars did all the innovative thinking and research. Authors should be prepared to explain the order in which they are listed and justify each authorship. The rule of thumb is the one who contributed most is listed first. This should be a joint decision of all co-authors, ideally made at the start of the study and reviewed periodically. Hollywood solves the problem in a different way. In conclusion, authorship should be based on the 4 ICMJE authorship criteria. All persons designated as authors should qualify for authorship. All those who qualify for authorship should be so listed. Gift, guest, and ghost authorship should not be practiced by authors or tolerated by editors, reviewers, and readers. And the order of authorship should reflect the true contributions of each author. Thank you very much for your kind attention, and have a good morning.
Okay, we now have time for an open forum, and I would like to start or get the ball rolling by asking Wang my first question. I was really impressed by your presentation. It could not help but remind me of the screensaver that PAASE is using. We have the San Juanico Bridge juxtaposed with the Golden Gate Bridge. And in a way, yeah, so if you, can you show that again, Juan? See, so that's the San Juanico Bridge and the Golden Gate Bridge. And PAASE is really a PhilAm community, as you can see. Now, whether by intent or serendipity, the first half of this session was composed of lectures from the other side of the world, except for us, which is down under, and nobody knows where that is. And the second half was composed of the three of us from UP Manila. And you did speak about the divide of the Pasig River between Diliman and Manila. But let me bring that further. What about between the US and other developed countries? As far as PAASE is concerned, for example, how would SIBOL be able to tap this resource, Wang?
Thank you, Joey, for that question and for showing us how you merge the two bridges. That's very significant. Actually, in one of the slides, I talked about mentors who've actually gone through the process of research translation. And if I'm not mistaken, one of them is actually in this session. He's one of those who's actually helping us, Dr. Serafica. And I'm talking about people like that who are helping us by mentoring us through the process. Because our learning, we're really just beginning to see how all of these things fall into place as institutions. I know that there are individual researchers who have been doing it and maybe are a little bit more knowledgeable than others. But as institutions, we need a lot of guidance and wisdom from people who've been doing this in environments which have been more encouraging over the past many years. So I think PAASE has a big role in this, and we look forward to working with the other members of PAASE and with the association itself. Thank you. You're muted, Joey.
Thank you. Thank you very much, Ewang. The floor is open to questions now. I have somebody with a private, it's in the process of being typed, I think.
So, may I ask a question or make a comment, Joey?
Yes, of course.
Yes, so it's nice. The bridging is not just between US and the Philippines, not just between UP Diliman and UP Manila, but also the bridging of basic biomedical research to research all the way to the clinics. But we like to start with what the medical doctors, the frontliners, are doing in a hospital. And that's what inspires us, basic research scientists, to do our work well, because eventually we want our research on products and services to be useful in the clinic. So I think Dr. Ewang highlighted the importance of starting to produce our local devices and supplies, because the healthcare in the Philippines, the system is just so expensive, almost unaffordable by the the people who are disadvantaged in life or do not earn a lot. So you're trying to come up with new devices, simpler, more complicated ones. But even the ordinary supplies in the hospitals. May I ask if all of those are imported, from the syringe to the needle, to everything, maybe except cotton? May we know from the doctors your range of reagents, supplies, and instruments and devices which are not imported?
Well, I guess I'll go first, Ma'am Giselle. First of all, the picture that I presented with the technologies and the different machines and equipment, that actually showed pretty much how much of almost everything is imported. I showed you a lot of, we as surgeons, once we get to the operating room, almost all the surgical instruments there are imported. And they used to be from the US, Germany, maybe Japan, but now we're seeing importation from China, from India, Pakistan. But we still have to see anything from the Philippines. Now, I think for the medical side, let's say you're talking about syringe and maybe even the stethoscopes, I don't think there's anything that's made locally. And that's why I raised the problem of are always not having enough, because we're always so dependent on these importations. And if there are machines, we buy expensive machines from abroad, and we're not able to take care of them when even small parts break up or we need replacement. We're not able to do it because we just don't have it here. We have to wait for the importation. We don't have the technicians who know how to fix it. So that's actually a problem.
So that's really one of my advocacies from way back, and I had spoken about it, Wang. So I'm so glad that you are a new champion and leader, to get more things done. So under the PICARI program that was funded through the CHED Philippine government, there were such projects to develop medical devices with the UC San Francisco, UC Berkeley system. Thank you, Ewan.
Thank you. Thank you.
We have a similar experience in our field. We recycle imported materials and fashion them into something else. For example, we cut off and refurbish syringes in order to create tracheostomas. We recycle silicone tubes in order to make trach ties and things like that. So and these are not SIBOL level technologies. These are very useful clinically, but they're not even things that would be patented. They're technology that we share and innovations that we share freely for people to adapt and use in their own settings. We have a question for Dr. Lapitan. Can you tell us what you do once you've identified a specific procedure which has an unacceptable rate of infection?
Thanks for that question. Well, that's really a challenge for the department. It's usually a multifactorial thing to have a surgical site infection. The first thing that we learned in the department, I don't know with the other surgical services, but since the start of this program, it's the awareness and the acceptance of the surgeons to own the problem. That is the first lesson to be learned. Actually, we're still in that process to have the surgeons accept that it is their responsibility to address and to find ways to find out the possible causes. I think the first thing that we usually do is take a look at the process of getting that wound and how that wound was dealt with. Usually how we prepare our patients for surgery, how the surgeons cut the wound, how the surgeons wash their hands. And that's why in the program that we set up, it's really how we take care of our wounds during the surgery and right after the surgery, and how we surgeons prepare for that surgery. That's what we review closely in those operations where we find a particularly high rate of infection. And that's what we see in our appendectomies, because these are done by fairly junior residents. So those are new to the team, and then done in the emergency room setup, done in patients who are off from the streets, off from wherever, in the emergency room without being properly prepared for that surgery. And I mean, cleaning the area of the area of surgery. So we're reviewing all these things and making sure that we address those gaps in our processes to lower our infection rates. So it's really a review of the entire process, which are unique for that particular operation. But also addressing, processes that are also present in all the other types of surgeries.
I have a question. For me, is gift authorship more common in certain cultures? And if so, would that not become very difficult to correct? That's a very perceptive question, actually. And we have written about the divide, if you want, between what we call collective versus individualistic cultures. I hesitate to use the terminology East and West or North and South, but those have been used also. Collective cultures are generally Asian, for example, while individualistic cultures are more Western, if you want, a stereotype. And it may seem at the outset that gift authorship is really more, not just common, but actually accepted in many collective cultural contexts. And this is because of the basic difference in perception of the self versus others. In the individualistic cultures, it is self before others, generally, like I introduce myself as Joey Lapeña, first name and last name, so self over family, of the UP, Manila, institution last. Whereas in many more traditional collectivistic cultures, I would be introduced in the exact opposite way. From the Philippines, University of the Philippines, the Lapeña family, Jose. Yeah, so Chinese names are an example. Last name first and first names last. Now, in that context, the connectivity of people to one another in collective cultures is considered to extend to the bond between mentor and mentee, between professor and students. And therefore, it has been argued, who am I as a new writer were it not for my professor? And how can I claim that what I wrote is just mine because I would not be myself without my professor? And in that regard, you will see it's very common for the so-called, what the West will call gifting, gift authorship, when in fact for the East, it is simply realizing or recognizing that I would not be who I am were it not for you. So that's an issue that is actually very much alive, and it is being discussed even today. The final answer is not available. The International Committee of Medical Journal Editors, which has drafted the criteria for authorship that I discussed, certainly tries to recognize that by leaving the criteria as open as possible for authors who wish to acknowledge, for example, their mentors and seniors and teachers by giving them a role in the conception, in the drafting, or at least in the revision, in the final approval, and in accepting responsibility. I'm sorry for the long answer, but I hope it addressed your question adequately.
I have a question. I have a question for Joey and then for Ewang. So, Joey, I wanted to know whether the requirement for the contributions of authors by many journals now would solve the problem to a certain extent. Second is, what's your position on equal first authorship? So recently we had a publication, but before we got it in, there was a controversy within my group. Two Filipinos, formerly my RAs, one already PhD, another one going for his PhD, and an American postdoc in our group. So generally, you know, you have big funding for multidisciplinary groups, and the three of them have this, you know, internal thing. I am supposed to be a first author, and then one of my PIs said triple authorship, first authorship is not allowed. But then again, you know, theoretically, philosophically, if the work was quite complex and each one really felt that he did something very significant, deserving of first authorship, then I, you know, asserted this idea that this is possible. And then we learned later that there are already procedures for this. So I wanted to know your position on this. I'll also ask Ewang's question, my question to Ewang. So when I was, you know, UP administrator, we really wanted to promote this multidisciplinary PhD and D programs, particularly focused on biomedical engineering. And at that time, PAASE was, well, because I was president in 2011. So Dr. Gobet Advincula and the University of Houston, they offered to develop a co-hosted biomedical MD PhD engineering program with the UP Manila. Okay. Then more recently, in the past few years, there have been many Taiwanese universities and linked hospitals interested to establish this, because I think that, you know, if you do not have the trained workforce from the bachelor's to the master's to the MD or postgraduate level, you cannot sustain, you cannot even begin to think of a biomedical engineering program to support the development of such instruments in the, you know, machines in the clinic. So Maybe Joey first and then Ewang.
Okay, thank you for your two questions. The first one is probably addressed by the fact that most journals have very stringent, excuse me, somebody's mic is not muted. One, we can hear a discussion in the background. Thank you. So most journals have very stringent requirements for disclosure, not only of conflicts of interest, but of contributorship of each author. And in many cases, authors are individually asked about the contributions of their co-authors as a means of journals checking for these different forms of misconduct. But there is really no perfect way of addressing this. And as an example, I have a private question, which is, I'll just read it, very delicate question. What would you do if you found out that you were deliberately removed as co-author in your advisee's paper? In the initial submission, my name was written, but the paper got rejected, only to find out later on that the paper was submitted to another journal and my name had been removed. This is not a new experience. It is something that many of us have encountered. I myself was once upon a time victim to this malpractice, if you want, or misconduct. Fortunately, there are ways of rectifying that. The Committee on Publication Ethics, or COPE, has many guidelines that you should be able to consult. You just Google COPE or COPE, and you will find everything from suggested steps to take in order for this to be rectified. At the best case scenario, the paper can actually be retracted if misconduct such as this is proven. Now, to the second question about co-first authorship, this is really a reality that is true in many cases. You may have equal contributions and you cannot write both names first. And so an asterisk is indicated after each name, shared first authorship. And the reason for that is really from the point of view of credit. Many universities and institutions require a first authorship for promotion. Never mind how important the paper was and that you were third or fourth author in a groundbreaking paper. What they're looking for is first authorship. So there is such a thing as co-first authorship. Unfortunately, not all journals allow it. So there are some journals that will say, no, our journal as a policy does not allow co or shared first authorship. My advice would be, if you need the first authorship, is not only to agree among yourselves beforehand, but to look for journals that will allow you to both be named as first authors. Ewang?
Okay. Yep. Thank you for that question. Si Ma'am Giselle is thinking like an administrator. So, ang tinatanong niya is a biomedical engineering program. Yes. Again, in one of the slides, I mentioned that all of these things have to be institutionalized. Otherwise, it can't progress. We can't progress based on the passion of a group of people or maybe a few alone. As long as we're not institutionalized, it's not going to move much further. And institutionalization, one of the ways is precisely the establishment of such a program. And I'm aware that there have been attempts to do it. And again, both the UP Manila and the UP College of Medicine is trying to push for such a program. So yes, thank you, Ma'am Giselle. And we're moving towards that. And we're hoping to be able to your input into such. Thank you.
I have a question.
Joey, sorry.
Yeah.
No, I was going to make a follow-up to that question I asked about the authorship. For example, the Japanese, writing papers is really just one part of work in, for example, a university setting. And there are a lot of other things that comprise being a university professor or instructor. So I'm reminded of papers churned out by very prolific Japanese universities, where always the head of the department is one of the co-authors, even if the name is at the very end. So that's an issue that must be considered, I think, right? Because it's not really just paper writing or the production of uh, scientific studies, but it's also you're there in the university or in that department doing many other things. So the the department chair, for example, the head of the entire orthopedic section of that prefecture is given a name because of the recognition of all the work that they've done for you as a member of that orthopedic community. So that's a follow-up comment lang to that question that I made.
Thank you, Ewang. We have a question for Mela. Quote, is such a program available in all hospitals? In many other countries, the MOH or DOH monitors infections among many hospitals and addresses these accordingly.
Yeah, thank you for that question. Actually, in theory and in paper, the DOH, particularly the PhilHealth, once you are PhilHealth, to be PhilHealth accredited, you must have some sort of a infection control surveillance, and part of that will be monitoring for surgical site infection. So that's supposed to be, in theory, should be in place in all, at least PhilHealth accredited, and and definitely all the DOH hospitals. But whether there is really a hospital driven, and even as you can see here in PGH, this is the program of the Department of Surgery. I'm not sure if the same program exists in other surgical specialties in PGH, but this is something that we are implementing in our department. But yes, indeed, in other hospitals, it is part, or in other countries, it is part of a program. In the Philippines, I know, like, for example, I know definitely in St. Luke's Medical Center, there is their infection control department or office. One of a team of nurses there are surgical site infection control teams that really follows up the patient, the surgical patients, monitors them, and documents all these surgical site infection instances. So there are programs probably in the major private hospitals. I am not aware of other such programs in the government hospitals, because it is a very labor intensive program, and for surgical site infection surveillance, a true program needs to be led by an infection control nurse, and these are highly specialized, highly skilled nurse. I mean, in our department, I just have an ordinary nurse who just has some additional training from our infection control nurse in the hospital, but she has developed the skills over the years that she's participated in the program. So it is a big effort. It has to be just recently we've presented this in the quality improvement committee of the hospital, but it is a very personnel intensive, and even skilled personnel intensive program. So it really takes effort and time to implement, but it should be worth it, and what we've seen is that it is possible with a little bit of push and effort, I guess. Thank you.
Thank you, Mela. I have a question on the YouTube chat group for Ewang. Hello po. Question for Dr. Wang, if ever po. The Office of the Vice President has been advocating for a shift to local manufacturing process to meet the demands of the COVID pandemic. Has SIBOL tried contacting her office to possibly tap into the OVP network to accelerate its outputs, such as looking for parts for prototypes, etc.?
Yes. Thank you again for that question, and thank you for bringing that up. One of the we during the start of the SIBOL COVID task force projects, we tried to contact many different offices, and I think we did try to contact the office of the vice president because one of the projects in the SIBOL COVID task force was the production of PPE, local production. And in order to move things faster and to facilitate the production, we heard kasi that the office of the vice president was actually already also thinking of doing that same thing, manufacturing. So we wanted to team up Tana with them. But beyond, I think, an initial call, there was no further follow-up. And I guess everyone was taken up by so many things during the COVID onslaught at the start. So there was no follow-up. But we'd very much like to work with that group again. They were actually tapping local industry, I think part of the local textile industry. And they were actually able to produce some PPEs, although not level 4. I'm not sure if they met level 3, but they were able to produce. On our part, in the SIBOL COVID task force, the project on PPE was, in a way, it wasn't shelved, but it was made not to address the immediate COVID needs, because we found out that we didn't have the materials and we didn't have the technology to be able to make at least the coveralls. So that project has gone into a longer term project for 2021. And we're probably going to work with PSHERD, which is another division of the DOST, looking into at least giving it more time to look into local manufacture of PPE, coveralls specifically, that will really match or will be good enough for level 4 PPEs. And by the way, the SIBOL has also taken the initiative to come up with a science policy forum, and eventually we will be holding a webinar on PPEs. So as you know, PPEs cover everything from masks to coveralls to your booties to your gloves. And there has been so much question and clamor from both academe and private sectors to learn more about PPEs and what they're made of, what kind of materials we need to make them out of, what's the licensing, what's the registration, licensing for both manufacture and licensing for importation. So we're going to be holding that. And I think that might be a big opportunity for us to work with maybe the Office of the Vice President also. Thank you, Joey.
Okay, so Joey, are there any more questions?
I'm receiving a lot of private messages, but there are many confidential issues being raised.
Oh my, so
yes.
Well then maybe you can convey them to the speakers.
I'm trying to reply in private. Yes.
It's now 10:47, so we're
can call it a day.
minutes beyond our planned time of this session. And I must say that I personally valued all the talks given by our speakers today. And well, just to emphasize the point again, that we cannot address health, medicine, disease, pharmaceuticals without covering the whole range of research, from the basic research to the medium advanced research, all the way to applications in the hospitals and the clinics. So I'd like to thank our speakers. Very deeply grateful for sharing with us your knowledge. And it's just telling you that in the spirit of PAASE, we are discovering many, many more valuable work that our researchers are doing in different parts of the world. And it really gives me great, great satisfaction and hope that eventually the Philippines will be able to catch up or ratchet up on S&T initiatives to provide Filipino people with greater quality of life through health, nutrition, and welfare. So thank you very much, everyone. And I hope you share, our viewers, our participants, share together with our speakers and our hosts the passion to pursue science and research. Wherever you are Filipinos of Filipino descent, and always give a part of your heart, your efforts for the benefit of the Filipino people. So thank you very much, everyone. Good day, good afternoon, good evening.
Thank you.
Bye-bye. Do we have another screenshot? So, Richmond, I'm really so happy about this session because yesterday we had our opening and there were some glitches. And then last night we met, Richmond and I, and then this morning you're in the briefing room with our speakers and of course our hosts. And so I felt that today was, better than yesterday. So thank you so much, including to Richmond. So he's our tireless webmaster and manager, the only one actually. So we're running the whole PAASE just on the efforts of our members. And then this guy, Richmond, he's a lecturer in a College of Arts and Letters and also full-time employee in Quezon Hall. And he's doing this for us because he believes in our cause. So thank you so much again, Richmond, and everyone who contributed to this session. Bye. And we should do this again, right? We're already liberated to be able to hold conferences whenever we want. Okay? Thank you so much.
Thank you, everyone.
Punch. Bye-bye. Nala, Ewang.
Thank you.
Russell.
Thank you very much.
To meet you, Russell. Thank you.
Bye. Nice to meet you.
Bye-bye.
Thank you, everybody.
Bye-bye.
Nice to meet everyone.
Conrad, okay, Star Wars mo. Mela, si Nilo Paner.
Sige, bigay mo sa akin, sir, email.
Yeah, that's very, ang galing ng ginawa mo. We need to do that also in ortho.
Yes.
Thank you, everyone.
Parang we don't want to leave, no? Parang we don't want to leave. Parang Mela, we don't want to leave.
Thank you, Joey. Thank you, Ma'am Giselle.
Alam po, ma'am. I'll be the last na lang po to leave. Sige po.
All of you.
Bye.
Bye-bye.
Thank you po.
Bye. See you.
Bye.
See you.
Thank you, sir. Thank you po.
Bye-bye.
Bye.
Thank you, Richmond.
Thank you.